inside a turbid scattering environment. It was used for detection of organic nanoparticles and tissues, for detection of milk components, detection of gold nanorods, and it was also extended for multi-layers analysis. It would. We also did a cooperation with the, Professor Michaeli and Professor Lalouche for detection of anti-leishmania particles. Professor Michaeli talked about it in her lecture using this method, and also for detection of penetration of nanodiamonds along different skin layers. Now we apply this nanophotonic techniques. You see, they're all nano elements for applying for sensing fishing nets in water. Now, what's in common for all of these applications that we did is that we want to sense the presence of elements that we cannot observe due to the scattering environment. This also happens in sea. In sea, the water, which seems clear for short distances, makes fishing nets almost, they cannot be observed easily, and we use the same technique, which is non-invasive. This non-invasive property of the technique we use for remote detection of fishing nets for underwater unmanned vehicles. So now let me introduce our technique. We call it the IMO technique, which is short for the Iterative Multiplane Optical Property Extraction technique. It consists of 5 stages and leads for extraction of the optical property of the reduced scattering coefficient. We analyze the phase, which is sort of lost due to the scattering, and from the phase analysis, we can extract these optical properties. So we start by illuminating a sample by a coherent laser, and we take several intensity images with a known distance between them. We insert these images into the phase retrieval multiplane Gershwin-Saxton algorithm. The output, I will mention for those who are familiar with the algorithm, is not the conventional output of the Gershwin-Saxton algorithm, because we take output from all of these intensity images. We take a phase image, which is actually a map describing the relative phase change accumulating along the path between the first and last intensity images that we took. Then we can separate this phase image into 2 different regions, multiple scattering and single scattering regimes, and we calculate the root mean square of each region. This is our quantitative indicator, which we compare with our theoretical model. From the comparison, we can extract a reduced scattering coefficient. Now, the technique is complex and full of details, which I will not discuss now. I can happily answer questions later. I will just state that our theoretical model describes the root mean square of the phase is a combination of 2 theoretical models, one describing the diffusion reflection, and it is combined with a phase model describing the differential pathing factor of the phase along the turbid medium. Let's take a look about our optical setup. This is the optical setup that we have in lab. You can see the laser illuminating the sample. The large spot that you can see of the laser on the sample, the opening, happens due to the scattering, which we want to analyze. You can see our camera taking pictures. a set of two perpendicular polarizers for optical clearing, and a lens in order to focus our images. And now you see that the camera, the lens, and the polarizer between them are all sampled on a moving stage with a motor that enables the distance between the pictures. Now the camera takes intensity images. It looks like this, and we usually insert them as an input for the phase retrieval algorithm. However, in order to apply this technique for fishing nets, we have to create optical magnification. That means to capture a larger field of view, and we do this by simply changing the distances between the lens and the camera, and the lens and the sample. You can see here how changing these distances actually applies zooming in or zooming out. We insert then these intensity images to the phase retrieval algorithm, and we get as an output a phase image. This phase image can be divided into two different regions. The single scattering regime, which is colored here blue, surrounded by the multiple scattering regime, which is red here. We calculate the root mean square of each region, and we can compare it with our theoretical model, as you can see here. This comparison allows us to extract the reduced scattering coefficient of our measured item. Now, some results. Actually, the first experiment we had to do in order to apply the technique for fishing nets was to adapt it to a different wavelength. The technique was designed following a red light wavelength for biological applications, and we first adapted the theoretical model and the experiment for the experiment that we're taking. You see by asterisks here for the blue wavelengths. I will mention our phantoms now. You can see on the left the phantoms that were measured for validating our theoretical model. These are 10 phantoms that we prepared with known optical properties. Every phantom has a different reduced scattering coefficient. The next step was to apply optical magnification without any zoom. Actually, I zoomed out here. You can see I adapted the theoretical model for magnification, and you can see by the asterisks the experimental results that fit with our theoretical model. Now, an important thing that we saw while magnifying our system is that our technique can be tailored for improved detection of desired specific ranges of reduced scattering coefficients. You can see here the gray areas are for different regions of reduced scattering coefficient. They shift with the magnification. Let's move on to fishing nets. Now, you saw this experimental setup that we had in the lab that all of these biological researchers were used on. We then replaced our sample with fishing nets. 6 fishing nets that were collected from fishermen we measured here. Then we upscaled the system, and we measured these fishing nets within an aquarium filled with seawater that we took from Tel Aviv, actually. The results show a highly significant difference between measurement of empty water and measurement of fishing nets. Let me show you. Remember this phase image that can be divided to different regimes? The red graph here is the multiple scattering regime, and it increases with the reduced scattering coefficient, where the center, the blue here, describes the single scattering regime, and it decreases with the reduced scattering regime. When we measured the seawater without fishing nets, we got results that fit with the with very low scattering, because we we got high value of phase RMS for the single scattering area and low value of phase RMS for the multiple scattering region. The fishing nets, in contrast, has the opposite behavior. We saw and we accept high phase RMS values for our fishing nets and low for our multiple scattering area, and low values for our single scattering area. So, okay, nice, we did it in the lab and aquarium, but now we want to apply it for actually measurements and see. But how? We started by trying to design an optical waterproof case for our system. We contacted with a research group from Haifa, and we designed an actual case for the system. You can see here the laser, the camera, and a bigger lens. And then we produced it. It came realistic. You can see here the closed package, and you can see also our experimental setup underwater after we applied some water resistancy tests on this setup. The next experiment will be conducted in a large pool that is connected to the sea, nearby here in Haifa. This presentation was maybe a little loaded, but I wanted to emphasize 3 points. One, the IMO technique can detect scattering elements within turbid media. We did some cooperation with other labs, and if anyone here was inspired for cooperating and searching and do research together, we can discuss it later. Second, our technique can be tailored for desired regions, for detection of desired region of reduced scattering coefficient. And third, we were able to take this nanophotonic technique and apply it for fishing net detection underwater, which will be in the near future applied in a large pool. Thank you very much.